Hello students, this is Professor C.P.S. Verma from Laureate Institute of Pharmacy. Today we will discuss the next chapter of your syllabus, that is volatile oils. We will discuss under this chemical test and classification of volatile oils and its extraction methods. Then first of all we will define what are volatile oils. The odorous volatile principle of plant and animal source are known as volatile oils. They are liquid. Volatile oils will evaporate or volatilize when exposure at an ordinary temperature, and so that they are called ethereal oils. They are also called as essential oil as the essence or concentrated constituents of the plant. General properties of volatile oil: they are soluble in alcohol, ether, or other liquid solvents, and practically insoluble in water. They are usually lighter than water. They possess characteristic odor, and they have high reflexive index. These are the general properties, and another next property. Both of them are optically active. They are secreted in special structures such as duct cells and Serogenous or lysogenous glands and trichomes. They are commonly found in species of Labiati and Rutaceae and Pyraceae, Gingiveraceae, Umbelliferaceae, and Myrtaceae, and Lauraceae. They are present in entire plant or in any part of the plant. Chemical nature: chemically they are derived from terpenes and they are oxygenated compounds. They are made up of isoprene unit C5 H8 and are usually mono sp or and diterpenes with empirical formula as C10 H16 for terpenes, monoterpenes, and then C15 H24 for sesquiterpenes and C20 H32 for diterpenes. Classification of volatile oils. Number one, volatile oils are classified as alcohol volatile oils, that is cardamom and coriander. These oils are coming under alcohol volatile oil category. Then aldehyde volatile oils, for example cinnamon, lemon peels. This will be under aldehyde volatile oils category. Ester volatile oil, lavender and mustard, and then hydrocarbon volatile oils, for example turpentine and black pepper. Then ketone volatile oils present in caraway and oxide volatile oil present in eucalyptus and chenopodium. Then phenolic ether volatile oils, for example fennel, nutmeg. Then phenol volatile oil, for example. Then preparation of volatile oils, how we will prepare. The principal method used in preparation of volatile oils from plants depend upon the distillation in water or steam. Then second, mechanical methods. There are some mechanical methods to extract or to take out the volatile oil, that is enfluence. This extraction of oil used in perfumery. Then Quayle method. Then extraction of solvent, enzymatic hydrolysis. For glycosidic volatile oils, for example mustard oil, we will use enzymatic hydrolysis. And then supercritical fluid extraction. This is the new technique by which we can take out the volatile oils from the. Then production and use of volatile oil. The total annual production of volatile oil is estimated to be in the region of 45,000 tons, worth approximately 700 million US dollar. There are about 100 commercially valuable volatile oils directly derived from plants. Then biosynthesis and chemical composition, we have already discussed the malonic acid pathway and for getting aromatic compounds formed by cyclic acid phenyl propanoid route. Phenyl propanoid route already we discussed. then C6H5, CH3. Then these aromatic compounds like this, the eugenol, chloride, enethol, anisole, and cinnamon, cinnamaldehyde, cinnamon oil, cinnamon. Then physical properties, although volatile oils differ greatly in their chemical constant, there are a number of physical properties in common. They possess characteristic odors. They are characterized by high refractive indexes. Then most of them are optically. Then the fourth point is their density is generally lower than that of water. The essential oils of saffron, clove, or cinnamon are the exceptions. So volatile oils are immiscible with water and they are sufficiently soluble in a part of their odor. And then aromatic waters are dependent on this slight solubility in water. Then volatile oil and fixed oil. Several points of differentiation exist between volatile oil and fixed oil. Volatile oils can be distilled from their natural sources. Volatile oils do not consist of glycerol ester of fatty acids. Hence, they do not leave a permanent grease spot on the paper and cannot be saponified with alkalis. Then volatile oils do not become rancid as do the fixed oils, but instead of exposed to light and air, they oxidize and rancid. Uses of drugs containing essential oil in pharmacy, the uses perfumery, food industry and food technology, miscellaneous industries as starting material for the synthesis of active principles of medicines, vitamins and fragrances. So there are different methods of preparation of volatile oils, distillation, measurement methods, extraction with solvent, enzymatic hydrolysis, supercritical fluid method, then processing and citrus roots extraction. Also, they are using volatile oil solvent to extract the volatile oil. For example, that is petroleum ether. We are using when the solvent penetrates the flowers and dissolves the natural perfume together with some waxes and other albuminous and coloring matters. This solution is pumped into an evaporator and then concentrated at low temperature. After complete evaporation of solvent, the flower oil is obtained. Then application, this method is used in perfume industry for the production of volatile oils using lipid solvents like petroleum ether or benzene. Advantage, it is quick, quality assured and economical. Then the decomposition of volatile oil is less as compared to hydrodistillation method. The solvent distilled off may be reused for extraction. Enzymatic extraction. Here the volatile oil is normally found in plant substances in the form of odorless glycosidal combination. However, the odori, odoriferous compounds are liberated free only by hydrolysis. Volatile oil of the bitter almond, that is benzaldehyde, then clove oil. There are different oil methods of production. It is prepared from the well developed flower buds by distillation method of analysis. Specific gravity of oil is 1.038 to 1.060. Refractive index is 1.527 to 1.53. Then cinnamon oil. Cinnamon oil method of production. Cinnamon oil is obtained from steam distillation of leaves and twigs of the plant Cinnamon cassia. It contains less than 1%. And the method of analysis, optical rotation, specific gravity, and refractive index are also these are also the parameter for Galactic oil. Then mentha oil, method of preparation, the flowering top of mentha collected and air dry. The dried flowering tops are introduced into a milled still where steam distillation is carried out for 3 to 4 hours under reduced pressure. In the first 2 hours, up to 85% of the oil gets distilled out. The density of the mint oil is less than water, because of which it floats on the surface of water. The distillate consisting of mint oil and water is introduced in a separating funnel where oil is separated by decantation. Then method of analysis, neutral to litmus paper, then optical rotation, specific gravity and refractive index are noted down, because these are the methods for standardization or analysis. Then clove. Clove, what is clove? Biological source of clove. It consists of dried flower buds of Eugenia caryophyllus, family Myrtaceae. Chemical constant, volatile oil 16 to 21%, phenol chiefly eugenol 80 to 88%, acetyl eugenol that is 10 to 15%, and alpha and beta caryophyllene chemical constant, and it also contains tannin 10 to 13%, pyrogalol, tannins, these are phenolic compounds, and other substances are methyl furfural and dimethyl furfural. These are the aldehydic volatiles. These volatile contents. Medicinal use, used as an antiseptic, used as a stimulant, used as carbonative, used as flavoring agent, and used as local anesthetic. This use is because of presence of use. When used as spice, used in toothache, used in dental preparation, mouthwashes, and oil used in perfumery. Clove oil and zinc oxide are used in temporary filling of diagnostic characteristic odor, aromatic, column small biconvex with rounded or triangular oil glands, fragments of parenchyma with the entire or portion of oil glands, then parenchyma, portion of loose parenchyma, then fibers, scleronomatous fibers associated with parenchymatous cells, then sclerites, oval to sub-rectangular, thickened walls having numerous simple or branch pits, then calcium oxalate is also present in the form of cluster crystals. And next is when starch is present, then it is from anthophylle, this is the mother clove. Then cinnamon, next fruit drug is cinnamon. Then cinnamon biological source, it is consists of dried the plant's inner bark of Cinnamon gelanicum. Then family Lauraceae, it consists of less than 1%. Volatile oil chemical constant, it contains 0.5 to 1% of volatile oil, 10%, 12% tannins, flavonoids, and the mucilage, calcium oxalate, starch, sweet substances known as mannitol. Then cinnamon oil contains 60 to 70% of cinnamic aldehyde, 5 to 10% of eugenol and benzaldehyde, and then cumin, aldehyde, and other terpenes. Uses carbonative, stomachic, milds, astringent, flavoring agent, aromatic, used as spice and condiment, then preparation of candy, dentifrices, denti, dentifrice, and perfumes. Oil is powerful germicide. The diagnostic characteristic, organoleptic characteristic, and all color, odor, and taste. You will have this color and odor and taste. These are the organoleptic properties. Then phloem fiber, stool cells, starch grain, calcium oxalate crystals, oil, and then next that is phenol. Biological source of phenol is Phoeniculum vulgare and family umbrella. The chemical concept phenol contains volatile oil 1 to 4%, fixed oil 9 to 12%, and protein 20%. The chief constant of volatile oil are phenolic ether and ethanol 50 to 60%, and ketone phencon 18 to 20%. Volatile oils also contains aniseed aldehyde, alpha and beta pinene, ascorbic acid, and niacin, etc. Then uses same like other volatile oils, carbonate, carbonative, stomachic, expectorant, flavoring agent, aromatic, stimulant, enthalmintic, useful in dental and mouthwash preparations. And then phenol water is useful in colic and flatulence in children. Then diagnostic characteristic. These are about the morphological characteristic, mesocarp, endocarp, endosome, and then VT. The next fruit drug is coriander, biological source. Dried drug consists of dried fruits of Coriandrum sativum, then family umbilical. Chemical constant, it contains volatile oil 1 to 4%, fixed oil 9 to 12%, and proteins 20%. The chief constant of the volatile oil are limonene, parasamine, then camphor and coriandrol, etc. Malic acid, tannin, and vitamin A. Then uses carbonative, stomachic, expectorant, flavoring agent, aromatic, stimulant, diuretic, enthalmintic, and aphrodisiac. Aphrodisiac diagnostic characters about the sclerenchymatous layer. Then under this, we'll discuss the endocarp, what is the endocarp and endosperm and VT again same like previous crude drug, phenol, same characteristic, then eucalyptus and then eucalyptus mentha. Mentha, synonym peppermint, biological source, it consists of fresh flowering tops of aerial parts of plants known as Mentha piperita and then family Lariaceae. Then chemical constant, the chief chemical constant of peppermint is volatile oil 0.7 to 1.5% and 6 to 12% of tannin. The chief constituent of volatile oils are menthol 50 to 60%, then methyl acetate, and then menthone and enol and small amounts of terpene. And these are the diagnostic characteristics of covering trichomes with then glandular trichomes. Then is also there dicite and then epidermis cell with wavy walls, negative factors, absence of calcium oxalate crystal in the mesophyll tissue. Uses carbonated stimulant, spasmolytic and anti